Adi. Adi. Okay. Hello, class. Hello, class. Good afternoon. Can you hear me now?
Yes, Doc.
Yes.
Can you hear me now?
Yes,
Yes
Okay, good
po,
afternoon
Doc.
to all.
Good afternoon,
Okay.
Doc.
Good afternoon, Doc.
Okay. So, okay, class. Good afternoon. Who is the coordinator here? Who is the coordinator for Histo? Okay. For paras. What's this? Oh, Histo. Okay. Okay, sorry. I will be fast for the previous lectures, just to repeat. Anyway, I time it up, we'll finish before 3 o'clock, done. Okay. Okay, again, the digestive system has two parts. We have the digestive tract, consists of oral cavity, esophagus, stomach, small and large intestine, anus. Ito yung mga parte na may mga butas, butases. Accessory glands, salivary glands, liver, gallbladder, and pancreas. Ito naman yung mga parts na may linalabas. Okay? So, this is digestive, not reproductive. Functions, you have ingestion. So, the food that you put into your stomach. You have mastication, putting it into smaller parts. Okay? And then a movement towards your stomach through the esophagus. You have the secretion mainly in the stomach, and then hormone release. Okay, here you have your accessory glands joining, and then you have your chemical digestion in your small intestine, absorption, small intestine, and elimination. Layers. So basically, this is a tube, and therefore you have layers. The layers are all similar except in the stomach. I mentioned that already, and in the esophagus. The layers of this gastrointestinal tract would be consisting of the mucosa, and the mucosa would have sublayers, lining epithelium, lamina propria, muscularis mucosa. And then the second layer would be submucosa, and then the third is the tunica muscularis, or the next is tunica adventitia or tunica serosa. Okay, so this is our example here. The first part, you will have your stomach, rather intestine, and in your intestine, you have here your mucosa. Okay, the muscles, submucosa, and then the muscles here. What is important to remember in the esophagus is that this is basically divided into three parts. You have the first third, which is wholly skeletal muscles, and therefore this is what you call the voluntary part. Okay, whatever you put into your buccal cavity, down to your pharynx and then your esophagus, first 1/3, that would be, you can still put it out, okay, or vomit it out. But the word vomit is basically used when it's already from the stomach. And then the second part would be mixed. Okay, skeletal muscles and smooth muscles. And the third part, which is next now to your stomach, basically This consists of smooth muscles. Okay. The mucosa. The mucosa, you have epithelial lining. And in the epithelial lining, you have variable, okay, depending on the function. Okay. So if it is basically protective, then you would know that you have what? Squamous, no? Rather stratified. Okay. Lamina propria is consists of your loose connective tissue rich in blood vessels, lymphatics, lymphocytes, smooth muscle cells, and glands. And then the muscularis mucosa, thin layer of smooth muscle. Now, what are the basic mucosal types? So when it is protective, meaning to say the thing that you put into your mouth, okay, initially, of course, in your oral cavity, pharynx, and then, of course, the esophagus, it is protective, okay, and in the anal canal, protective. In the secretory is basically your stomach. Absorptive, the small intestine, and the absorptive and protective is your large intestine. Okay, so you would see here, this is protective because you have here stratified, no? Okay, and here in the secretory, you will find here basically long, diba? Columnar without goblet cells, and so on. Here you have your villi, okay, and here you have your large intestine. Okay, we'll discuss it more. Okay, so the lamina propria penetrates the core of intestinal vilus. From here, the lamina propria, from here up to this one here, huh? Okay, this one here up to this one. Penetrates the intestinal vilus with microvasculature and lymphatics called the lacteals. These are your microvasculature, wherein you find lacteals, okay? You find the lymphatics called lacteals. Submucosa. What do you see in the submucosa layer? Okay, this is basically dense, irregular connective tissue with larger blood vessel, lymph vessels, and the submucosal or Meissner's plexus. When we have in the examination, you do not just place, if it is, what do you find, okay, in the submucosal, you do not just say submucosal, okay, plexus, you have to put the name, if there is a name of the glands or of the plexus. And then this may contain also glands and lymphoid tissue. Again, if there is already a name in there, say the name. Do not just say submucosal glands. Nerve plexus. Okay, the submucosal plexus or Meissner's plexus controls secretion, okay, located in the submucosa. So you cannot say submucosa, nerve plexus, say Meissner's plexus. Tunica muscularis, or the other name for this is Muscularis Externa. Smooth muscle cells organized as two or more layers. Okay, you have the inner circular and the outer longitudinal or ical. Again, you have your nerve plexus, myenteric plexus, or Auerbach's plexus. This controls the movement located in the tunica muscularis layer or between the circular, okay, between the circular and the outer longitudinal layers. Tunica serosa or adventitia. You call it tunica serosa if the thin layer of loose connective tissue rich in blood vessels or lymphatics and adipose tissue with simple squamous epithelium or mesothelium. If there is no mesothelium and or you don't see any simple squamous, then you call it tunica adventitia. Oh, you have taken this one. This is just to show to you the different layers. You have the mucosa, okay. You have your submucosa, and then you have your tunica muscularis, and then you have your serosa or adventitia. Esophagus, then the layers in the mucosa, you have your what? Stratified squamous non-keratinized epithelium. The lamina propria is loose connective tissue or loose lymphatic tissue plus superficial esophageal glands. And then you have your muscularis mucosa, which is smooth muscles. The submucosa here you have in the submucosa, this here, you find here your deep esophageal glands, okay? Okay, deep esophageal glands. Also take note, class, that in the esophagus, you may see anterior esophageal glands, meaning to say the first part of the esophagus. And you might find also posterior, okay, esophageal glands in the area in the last third portion of the esophagus. But basically, you find in the submucosa deep esophageal glands. So you have to say deep. You do not just say esophageal glands. And the muscularis externa, of course, you have here your okay, inner circular and outer longitudinal. And then here you have your tunica adventitia. Okay. The stomach. Okay. So again, you will have the different layers. Okay. Now you see here longitudinal folds or rugae. So these are longitudinal folds or rugae, and this would be your gastric, okay, gastric pits. And then you have your submucosa, and then your muscularis externa, and then tunica serosa, okay? So what is the lining epithelium here? Lining epithelium, simple columnar without goblet cell. Plus you have to put without goblet cell. Without it, is you just put a simple columnar, then it's wrong. Okay, the lamina propria, you have loose connective tissue, loose lymphatic tissue, and gastric glands here. Okay. Area here, and there. Area here, and there. And then you have your submucosa, the muscularis mucosa. Okay, this would be your muscularis mucosa. And then you have in the, as I mentioned, you have dense connective tissue and Meissner's plexus. In the muscularis externa, you have 3 smooth muscles, not 2. Inner oblique, middle circular, and outer longitudinal. And then you have also your Auerbach's plexus, okay, between the 2, okay, in the muscularis externa. And then the last is serosa. okay, because it is covered by visceral peritoneal layer, and therefore you call it tunica serosa, or the lining is the presence of mesothelium. There you are, the mucosa. You have the submucosa, you have Muscularis Externa, and the serosa. Okay, this is to let you know that when you examine parts of the stomach, you would see different structures. They are not all the same. Okay, which is important in the uh, secretions that we could find in this particular area. Okay, so you have your stomach here. Take note. You have here this one. Okay, here. You will see you have your surface lining cell. So surface, okay. So they secrete first there, surface, okay. Because if you take something that is harmful to the surface, then this will be the one that would be, react. And next you will have your, okay, regenerative cell. This is just in the word regenerative, it would mean that if the parts of this are harmed, then the regeneration starts from this area. And then you have your mucous neck cells. Certainly it has its function. And then the oxyntic, okay? And then you have the cymogenic and enteroendocrine cell. We will discuss more in succeeding slides. The gastric glands. So the mucous neck cells, of course, secretes mucus. Your chip cell or cymogenic cells, pepsinogen. Your parietal cell or oxyntic HCl, an intrinsic factor. Enteroendocrine cell, the argentophil cell, secretes serotonin. Here, you find this all in your textbook of Junquera. Gastric glands, again, the cardiac region. This is not part. This is now part of the stomach. You find no. You don't find chip cells and few parietal cells. In the pyloric region, you find the short gastric glands, no chip cells, also few parietal cells. In the punctic region, all cells are present. Okay. Here, you find here your mucous neck cells. Okay, that is the gastric pit. Okay, and then you have your mucous neck cells, and then you will have your principal cells, and then you have their oxyntic or parietal cells. And of course, you have here your muscularis mucosa. Okay, so in other words, these are the areas that is part of, that is you label layer of the mucosa. Okay, again, okay, that would be your principal cells. Okay, then you have your parietal or oxyntic cells. Okay, take note of the color. It's an E. Okay, staining. And you have here the nucleus, thin. Cytoplasm is basically clear. Now compare it with your parietal. Okay, okay, it's not clear, no? Site where the digestive processes are completed and where the nutrients are absorbed. This is the small intestine in the duodenum, jejunum, and ilium. Here. So of course, By the way, have you taken the digestive system in your anatomy, President? How? Have you studied already the digestive system in your anatomy? Anyone please answer.
Yes, do.
Yes?
Yes, Doc.
Let's go, Doc.
Okay, so basically, you know this, okay, even the length of each part, duodenum, the jejunum, and the ilium, okay? Okay, what are the structural specializations that you see in your small intestine? You find the Plexus circularis or the valves of Kerckring, okay? These are permanent transverse folds that contain a core of submucosa, circularly arranged folds. And then you find the villi, short finger-like and leaf-like, depending where it is located. Projections in the mucosa that extend into the lumen, principal site of absorption of the products of digestion, 0.5 to 1.5 millimeter in length. There you are, okay? There you are, the intestinal villi. We say it is finger-like, and we say it is leaf-like. Microvilli, tightly packed microscopic projections of the apical surface of intestinal absorptive cells, give the apical portion of the cell a striated brass border appearance. So when I ask you what is the appearance of the microvilli that you see, for example, in the practical, then you would say it is striated or with brass border appearance. Okay. Otherwise, if we are asking for the lining epithelium, I'll be very specific. Okay. There you are. Okay. Increase, of course, all of this would increase the mucosal surface area. Okay. The absorption. Plica circularis increases the absorption by 3 times. The villi 10 folds, and microvilli 20 folds. A total of absorptive area of greater than 200 square meters. There you are, okay? The villi, okay? Now, the crypts of Leverkusen or intestinal glands, the enterocytes you see, it is columnar cell for absorption. The goblet cells which secrete mucin, secreting glands, okay? And then you have your Paneth cells, okay? These maintain innate immunity, innate, sarili, mula na tayong pinanganak, ha? Innate immunity by secreting antimicrobial substances. And then you have the enteroendocrine cells, produces hormones like secretin, mutilin, argentophil cells. Okay, there you are. The Paneth cells, location is in the basal portion of the intestinal crypts. Okay, they are exocrine cells with large eosinophilic granules in their apical cytoplasm. So this is very distinctive. Granules release lysozyme, phosphorylase A2, and hydrophobic peptides called defensins. Okay, now these are the cells, okay, that you find in the small, okay, intestine, enteroendocrine cells. And you see the different cell types and then the hormone produced, okay, secretin, mutilin, cholecystectonin, and serotonin, and the action of which are the secretin, pancreatic and biliary bicarbonate and water secretion. Its It inhibits gastric acid secretion, stomach emptying. Mutilin increases gut motility. Cholecystokinine, pancreatic enzyme secretion, gallbladder contraction, and inhibits gastric acid secretion. Serotonin increases gut motility. Please take note of this. Now, the crypts of Leberkuhn. Okay, there you have your neuroendocrine cells. Okay, like no, you will notice that when it is uh, relating to the neurons or the what you call the in the nervous tissue, you would see that they are lighter, no? Enterocytes, long or columnar, okay. Goblet cells, white around, okay, so that would be your goblet cells, secretion mucus. And then your panet cells. This is your panet, huh? You see there, red, huh? Naka-lipstick ito, huh? Okay, so these are some of the cells that would be as in the practical. The M cells, they are specialized epithelial cells in the mucosa of the ileum, in the ileum only, overlying the lymphoid follicles of Peyer's patches. So if you see their Peyer's patches, on top of it, overlying it would be your M cells. Okay. EM cells, these are modified intestinal epithelial cells that assist in antigen presentation by conveying micromolecules from the intestinal lumen to underlying compartments, okay, of the lymphocytes and macrophages. So this is the basal. It is basal membrane invaginations containing many intraepithelial lymphocytes and antigen presenting. Selectively endocytose antigens and transport them to the underlying lymphocytes and dendritic cells. Dendritic cells, again, origin is nervous, no? Dendrites. Nerve plexus, again, you have your Meissner plexus, control secretion, located in the submucosal layer. Nerve plexus, okay, myenteric plexus or Auerbach's plexus, located in the tunica muscularis between the circular and longitudinal layers. Duodenum, okay. The mucosa is simple columnar with goblet cells. While in the stomach, you have simple columnar without goblet cells. So in the intestine, up to the large intestine, you will see there with goblet cells. Lamina propria, muscularis mucosa. The submucosa, presence of Brunner's gland. Okay, these are your Brunner's gland. There you are, no? Okay. Described as branched tubular mucous glands seen in proximal duodenum. Okay, proximal part of the duodenum. And you know that duodenum is very short. Okay, proximal part, alkaline. Okay, secretes alkaline, which neutralizes chyme. So, duodenum. Okay, take note, this is always given in the practical Brunner's gland. Identify it as a Brunner's gland, and then the question, where do you find this in the submucosa? Or what important structure you could see in this layer? So you say Brunner's gland. Tunica muscularis, again, back to two smooth layers, inner circular, outer longitudinal. And of course, last is tunica adventitia. Jejunum, okay, the same, simple columnar with goblet cells. Lamina propria, Muscularis mucosa, submucosa. Okay, there is no name there that you find gland, no? Tunica muscularis, and then again the eye call, inner circular, outer longitudinal, and then your tunica serosa. Okay, tunica serosa. There you are. Ilium, very important. Mucosa, simple columnar again with goblet cells. Lamina propria, here you will find well-developed mucosa associated lymphoid tissue or MALT, consisting of large lymphoid nodule aggregates known as Peyer's patches. So when we point out to you this here, okay, you find there, you do not call it only as malt, but you say Peyer's patches there, Peyer's patches, okay. Muscularis mucosa, there, and then you have submucosa. Again, there are no glands in the submucosa, only in the what? Brunner's gland in the duodenum. Okay. Tunica muscularis, the same. Okay, so it's easy. Now, here you will have to differentiate those structures that you may see in two or in all of these small intestine parts. So, plica circularis, description of it. Okay, you have numerous, well developed in the duodenum. Okay, less numerous, well developed, and in the ilium, less numerous, less well developed. In your intestinal villi, again, leaf-like, okay, finger-like jejunum, and club shape, okay, finger, and then next one is naglumalapad, leaf-like, club shape, okay, club. And then your goblet cells, few in the duodenum. As you go further, numerous, and ilium more numerous. And even in the large intestine, it is very, very numerous. Submucosal gland, in the duodenum, you find Brunner's gland. In the jejunum, none, but rather numerous, but absent rather. In the submucosal glands, absent. So you find only in the Brunner's gland in your duodenum. Now, the lamina propria, dense, okay, are the same all throughout, but when it is already ilium, you do not describe them as dense or nodular. You say Peyer's patches. The length, okay, so you have 10 inches in the duodenum, 8 feet. In your jejunum, ilium is 12 feet. Okay, the width, the widest, okay, duodenum. Why? Because so many secretions coming from the accessory, okay, goes into that opening through, to the duodenum, and narrower and narrower. And then your mesentery, absent in the duodenum, present in both the duodenum and ilium. And the mobility, non-movable, very short, no, attached. And then you have the two very movable. Remember, okay, class, presence of gland in the submucosa seen only in duodenum, and the name is Brunner's gland. The villi tend to be longest, okay, mahaba sa duodenum, and become shorter and shorter as you reach your ilium. And then you have your lymphoid tissue becomes more prominent in the ilium and is relatively absent in the duodenum. Proportion of goblet cells increases distally. So you have goblet cell in your duodenum, pero more as you go distally up to the large intestine. Okay, now characteristics of the jejunum and ileum. I know you have studied this already in anatomy. Okay, so we'll go to other part. The Plexus circularis, you know this already, large, tall, and closely packed. There you are if you dissect it. The proximal ilium, okay, low and sparse. Okay, distal ilium, there is no more plica circularis. The vasa recta, ilium, the vasa recta is shorter and more complex arterial arcades. Okay, while in jejunum, it is long but not complicated or not complex. Okay, so this will be basically we ended up on that particular small intestine. We go now to the large intestine. Principal functions: reabsorption of electrolytes and water, and elimination of undigested food waste, feces. Divisions: cecum, appendix, colon. You have your ascending, transverse, descending. You have your rectum. You have your anal canal. There you are in your anatomy, you know that. Mucosa, okay, penetrated throughout its length by simple tubular intestinal glands, lined by goblet and absorptive cells with a small number of enteroendocrine cells. This is the mucosa. The columnar absorptive cells have irregular microvilli and dilated intercellular spaces indicating active fluid absorption. So when you see the microvilli to be dilated, indicates active fluid absorption. The goblet cells are more numerous. Lamina propria is rich in lymphoid cells, lymphoid nodules that frequently extend into the submucosa. Richness in malt is related to the large bacterial population of the large intestine. Tunica muscularis has longitudinal and circular layers. Parehas din, okay? Fibers of the outer longitudinal layer gathered in three separate longitudinal bands called tinea coli. There you are. Intestinal glands composed of two types of cells. You have columnar cells, the first one. You have columnar cells, okay. And then you have your, these are absorptive cells. And then you have the mucosecreting goblet cells, okay. Columnar, basically here, absorptive. The area, top, like the villi of the small intestine. Okay, now, muscular wall is relatively thick and capable of powerful peristaltic activity, composed of inner circular, outer longitudinal layers. Okay, the outer longitudinal layers forms longitudinal bands called tinea coli. Okay. Outer serosal surface, okay. Tinea coli are there. Haustrations. There you are. But this is better here. Haustrations. Appendices epiploica. And then the inner mucosal surface. Semilunar pulse. Semilunar, not like in the small intestine, it is, diba? Circular. Here's semilunar. The colon, okay? You will see here your mucosa, okay? And then you will have your simple columnar with goblet cells, lamina propria, muscularis mucosa, submucosa, there you are, tunica muscularis, and then again ichor, and then tunica serosa. Mucosa, appendix, the same, simple columnar with goblet cell, lamina propria, lymphatic nodules. They have no names in the appendix. Okay, lymphatic nodules. Muscularis mucosa, submucosa, the same, no? Now, class, if the practical you see one whole slide, this one you can see the entire specimen. Think always of appendix. Okay, so you have your mucosa here. Okay, you have your lymphatic nodules, but no name. Enough to say lymphatic nodules. There you are. Anorectal junction. This is very important, class. Okay, now simple columnar epithelium in the rectal. Okay, simple columnar epithelium. Now, in the junction of it, okay, you will have stratified squamous, non-keratinized. Remember stratified squamous? You have also that in your what? In your esophagus, no? And then you have muscularis mucosa disappears. You don't find any more muscularis mucosa. That would signify the anorectal junction. The circular layer of tunica muscularis, okay, circular outer, diba? Circular, inner, circular, outer longitudinal. The circular layer of tunica muscularis thickens to form internal anal sphincter. Okay, that is your circular. Now, the external anal sphincter is formed by the striated muscles of the perineum. Striated muscles. You know that in your anatomy. Now, these are the diseases that you can find, okay, detect in your, that may occur in your gastrointestinal tract. Celiac disease, celiac or sprue, disorder of the small intestine mucosa, intestinal mucosa, that causes malabsorption and can lead to damage or destruction of the villi. This is an immune reaction against gluten or other proteins in wheat or grain, okay? And resulting inflammation affects the enterocytes, okay, the enterocytes, leading to reduced nutrient absorption. So, celiac disease, this is a malabsorption caused by damage to the destruction of the villi, okay? It's basically an immune reaction to gluten or other proteins in wheat or grain, resulting to inflammation against the enterocytes, leading to reduced nutrient absorption. Crohn's disease, celiac or sprue, and then we have Crohn's disease. Chronic inflammatory bowel disease that occurs most commonly in the ileum or colon, resulting from combination of immune, environmental, and genetic factors. Excessive lymphocytic activity and inflammation can occur in any or all layers of the wall, producing pain, localized bleeding, malabsorption, and diarrhea. Colorectal cancer, adenocarcinoma that develops initially from benign adenomatous polyps in the mucosal epithelium, more common in individuals with low fiber diets, which reduce the bulk of fecal material, prolongs contact of the mucosa with toxins in the feces. You see this by doing sigmoidoscopy or colonoscopy, and then you do a fecal occult blood. Now, we shall continue now with the organs associated with the digestive tract. Okay. Okay, you have your major salivary glands, sublingual, submandibular, parotid. This will be discussed by Dean Lin, including the pancreas. Liver and gallbladder, I will be discussing this one, no? The products of these organs facilitate transport and digestion of food within the gastrointestinal tract. All of these, salivary, pancreas, liver, and gallbladder. And you have taken this in your anatomy, as you said, and of course, you know now all this part. This is the pancreas, okay, this is the duodenum, and then your liver, right lobe, and then you have the gallbladder underneath your liver. Liver, it is the largest internal organ of the body, 1.5 kilogram or 2% of body weight. Bile is complex substance required for the emulsification, hydrolysis, and uptake of fats in the duodenum. So you have your right lobe here, your coronary ligament, coronary, nasa taas as a crown, and then you have your left lobe, you have here the falciform ligament, and then ligamentum teres. That's more in the laboratory. And then you have your caudate there, the caudate part, and then you are down here, your, okay, other part, no? This is your gallbladder. We are interested in the histological part. Okay, so this would be your liver, a part of the liver, and you see this, the portal area or triad. When you see triad, of course, you would know the portal vein, okay, and then the hepatic artery, right there, and you have the bile duct. So you have a duct, an artery, and a vein, triad. Okay, you have in this central part of the lobule, you have your central vein. Okay, okay. These are your lobular boundary. Okay, so there you are. Hepatocytes, these are the cells in your liver. Synthesis and endocrine secretion into the blood are the major plasma proteins function of the hepatocytes, including albumins, fibrinogen, proteins, and then transferrin and many others. You have your process of gluconeogenesis formation, or you have your storage of your sugar, detoxification and conjugation of ingested toxins and their functions, no? Storage of glucose in glycogen granules and triglycerides in small lipid droplets. Storage of vitamin A, removal of effects, erythrocytes, ineffective or quite old erythrocytes, storage of iron in complexes with the protein ferritin. Okay. Hepatocytes. It is large cuboidal or polyhedral epithelial cells. Cuboidal or polyhedral, so many sides. It is large, round, it has large central nuclei. Okay, okay. And then the cytoplasm is eosinophilic, rich in mitochondria. It has so many functions to do. That's why it is rich in mitochondria. And then it is binucleated. Okay. The liver is slide in the histology wherein you do not forget. You see that at the beginning, you see that at the middle, you see it in the end. The liver cells. Okay, now this is stained by eoxine, hematoxylin, eosin. Hepatic sinusoids penetrated endothelial cell. So this is your central vein. Okay. And then the perosinusoidal space, you call this space of Bise. Okay, so when you say sheets of hepatocyte, that is the arrangement of your hepatocyte sheets, or what? Oh, you should know the different description, no? And then the sinusoids are this one, the white ones, no? Spaces. Okay. Portal triad, okay? Venial branch of the portal vein with blood rich in nutrients, okay? But low in oxygen because it is a vein or venule, therefore it is low in oxygen, but it is rich in nutrients, okay? Now, arterial branch of the hepatic artery that supplies, okay, your oxygen. So basically there are two, okay, because one is poor in oxygen but rich in nutrients, and the other one is rich in oxygen. One or two small bile ductules or cuboidal epithelium branches of the bile conducting system. Okay, here you are. Okay, so this will be your portal triad. You have a venule, you have an arterial branch, and you have a duct. But so Cooper cells, Cooper, hindi Cooper. So anyway, Cooper cells, stellate macrophages, stellate, ibig sabihin Strelia, star-like, with the sinusoidal lining. These are the sinusoidal, okay, sinusoidal lining, okay. Detect, the function of which is detect and phagocytize effect, no? Again, it is ineffective or old erythrocytes. They are also antigen presenting cells, okay, like your M cells in the intestine, okay. Remove bacteria and debris in portal blood, okay, portal blood. Okay. What are E2 cells? E2 cells are hepatic stellate cells. Okay. Hepatic stellate cells. It is in the perosinusoidal space. Small. It stores vitamin A and other fat-soluble vitamins. Produce extracellular matrix components, becoming myofibroblasts after liver injury. So this is important in the regeneration. Myofibroblasts after liver injury, and do cytokines, release cytokines, produce cytokines that help regulate Cooper cell activity. So take note of the E2 cells and the Cooper cells. Okay. Okay. The bile canaliculi, this exocrine secretion of bile, release of bile. Bile canaliculi form a complex anastomosing network of channels through the hepatocyte plates that end near the portal triad. Empty into the bile canals of herring composed of cholangiocytes. Here, okay. So this is now your bile, okay, ductule. You have the bile canals of herring. Okay, bile canals of herring. This one here. This one here. And then you have the cells lining. Okay, the ductules, you have your cholangiocytes. Okay, these are hepatocytes. Okay, the lining are the cholangiocytes. Okay, and these are your what? Your uh, bile canals of herrings. Okay, so this is now your lobule. Okay, the lobule, your liver. Okay, so you have now here what? You have here the central vein. Okay, you find in the center of the lobule. Okay, okay, and then at the periphery, at the areas where it is 1, 2, 3, 4, 5, 6 hexagonal, no? You have your triads here, okay? And then, of course, these are the plates of your hepatocytes. And this one here, of course, you find the bile canaliculi, okay? This is the bile duct, receives bile from the canaliculi, there, from these canaliculi, of course. And these are the fenestrated lining or endothelial cells of the sinusoids. Of course, this would be your what? Portal, okay, arterial. And the blue one would be your portal venial. So you see the portal, the triad area, okay, around this lobule. And this is your central vein. Now, structure and function of the liver. RER, rough endoplasmic reticulum, synthesizes, okay, synthesis of plasma proteins and causes cytoplasmic basophilia. The smooth detoxification of substance in blood which are excreted with bile. And then you have your glycogen granules, small lipid droplets, hemosiderin, temporary storage of glucose, glycogen, of triglycerides, small lipid droplets, and hemosiderin of iron. Peroxisomes, oxidation of fatty acids, Golgi complex, synthesis of plasma proteins and bile components, and mitochondria, energy. They needed to activate all these functions of the liver. The concepts of structure, function, relationship in the liver. Classic lobule. Classic lobule emphasizes the endocrine function of hepatocytes as blood flows past them toward the central vein. That is classic lobule, which we have been discussing earlier. Portal lobule, exocrine function, and the flow of bile from regions of 3 classic lobules, okay, toward the bile duct in the portal triad of the center. So, pa-outward siya, no? Okay, hepatic asinus emphasizes the nature of the blood supply to the hepatocytes and the oxygen gradient from the hepatic artery runs to the central vein. So, classic endocrine function, portal lobule exocrine function, hepatic asinus emphasizes the nature of the blood supply to the hepatocytes and the oxygen gradient from the hepatic artery. Please study this, okay? Read your textbook for better understanding. This is very important. Okay, again, this is an illustration of your classic hepatic lobule. Okay, it's one. Okay, you have here your hepatic arteriole, bile duct, portal vein. Okay, the triad. Okay, this is your central hepatic. Okay, central vein, or hepatic vein here. The portal lobule drains bile from hepatocytes to the bile duct. Okay, drains bile from the hepatocytes. These are all supposedly these are also hepatocytes to the bile duct. There you are. This one. There you are. This is your portal. Okay, lobule drains bile from the hepatocytes to the bile duct, from the central vein going towards the portal triad. Okay, there you are. Now, this one, the portal asinus, the portal asinus, this would be your portal asinus, supplies oxygenated blood to hepatocytes. Okay. Now, there are three zones. Okay. The zone, that zone 1 is mostly oxygenated. Okay. Zone 2 would be, uh, less oxygenated, less oxygenated, and then you have your portal 3, which is the least oxygenated. Okay, zone 1, is more oxygenated, zone 2 lesser, and zone 3 is the least. Okay. Okay. The biliary tract and the gallbladder. The bile produced by the hepatocytes flows through the bile canaliculi, bile ductules, and bile ducts. So canaliculi is smaller, ductules becomes bigger, bile ducts where you secrete where the bile passes. These structures are gradually merged. They gradually merge, forming a converging network that ultimately forms the common hepatic duct that joins the cystic duct, okay, from the gallbladder and continues to the duodenum as the common bile duct. Okay, you have here your cystic duct from the gallbladder. You have the common hepatic duct from the liver. They join to form the common bile duct, which enters into the duodenum. Okay, where you would find the one the controls is the sphincter of Oddi. Okay, and they pass through the major to the intestine, the duodenum, to the major duodenal papilla. Okay, so you know this in your anatomy. Okay, please review. Okay, this one here. Now, the gallbladder. Okay, you have here your mucosa. Okay, and then the muscularis, muscularis, and then adventitia. Okay, the gallbladder is described as a hollow, pear-shaped region, 30 to 50 ml of bile, and the lining epithelium is simple columnar epithelium. Okay, simple columnar epithelium. That is your gallbladder. Okay. It is found behind your right liver, no? Behind, inferior, okay, to the liver. This is your pancreas. Now, this is the gallbladder, okay. Okay, you have your mucosa here, okay. Okay, this part, okay. You are You do not have here the muscularis. You have lining epithelium, lamina propria. Next are the fibromuscular layer, and then the perimuscular layer, tunica adventitia, and tunica serosa. Now, as you see, this is attached to your liver. So you find here your, when it is okay, depending on its attachment, the fourth layer would be either tunica adventitia or tunica serosa. Okay, there you are. This is your invagination of your mucosa. Invaginations, and this is what you call the RA sinus, Bruchitansi Ashop sinus, RA sinus. Okay. Here the mucosa. Okay, so we ended up with our gastrointestinal tract, okay, from the esophagus and then down to your anus and anorectal junction up to the liver and your gallbladder. Okay, class. Okay, review. Okay. Okay. Okay, class. How? Yeah. Could you hear me? Class, could you hear me? Hello.
Yes po, Doc.
Yes, Doc.
Yes, Doc.
Talagang clear. Wala tayong disturbance ngayon, ano? Okay, good. Now, would you kindly get 1/4 sheet of paper? It is easy, no? I reviewed the gastrointestinal tract. I repeated it because that was a very bad time. The signals are so bad that I have to repeat it. Now, would you kindly get 1/4 sheet of paper? Okay, class? You have all now your 1/4 sheet of paper? Answer me. Apurilio. You have already? Apurilio. Uy.
And here po, Doc.
Okay na, you have already 1/4 sheet of paper. Ilasyon. Meron na. Sino pa ba itong mga Pixon? You have already. Okay. And then you have a chew. You have already. And the others that are, well, good results in the first semester or whatever. Anyway, this is a yearly subject, no? Okay, please answer the following, no? To the best of your ability, okay? If you cheat, it's up to you. You know, not, I'm sure you are not copying because you are, well, if you might be 2 or 3 in your area, so you can, okay, ready, class? President. Sino bang president dito? Huh? Uy. Ready? All of you now?
Ready, Doc, ready.
Yes,
Okay,
Doc.
please answer me. Of course. Okay. Ready. Okay. Honesty is the best policy. Okay. Okay. Please try your best. No, this is a test on your retention. At the same time, this will lead you to know the important questions that I may ask, and even in the practical. Okay, class? Okay. Okay, let's start. Okay, identify the organ. Yeah, baka kayo nagkukopya, tumitingin sa libro. Anyway, what is the lining epithelium? Lining epithelium, ayan, klarado, diba? And then identify whole structure, pointed. Next, characteristic cell. What is the characteristic cell of this organ? Okay, ang dali nito. Identify that, okay? Identify that portion, okay? Identify that structure. Oh, diba? Lining epithelium is what? This would be your lining. Ano yan? Okay. Nasa protective tayo. So it is whatever. Okay, this one. Okay. Lamina propria. Okay. And your, what are these? And that one. Okay. Done. Next. This one. Okay. Ano, muscle yan. Okay. Next. Oh, identify according to the arrows. Okay. Identify the organ. Identify the structure. Para may idea kayo pag practical. What layer? Okay, is this? You find this, identify the structure. Identify organ. Identify structure. Okay. What layer is the identified structure? Okay. So you should know your layer. Saan ba yan? Is it in the submucosa? Is it in the tunica muscularis? Whatever. Next. Identify organ. Oh, madali yan. Identify structure. And then again, identify the organ. Okay. Identify the structure. Okay. Next. Yan. Identify organ. Characteristic cell. What is that characteristic cell? Identify organ. Characteristic cell. Those are in the intestine, gastrointestinal tract. Intestinal glands. Okay. Identify cell. Ayan. Pag hindi pa naman nakuha yan. Identify cell. Ayan. Si Miss, the function of the first cell. And then ito yung may lipstick. Okay. Function. So, there you are. Oh. Identify organ. Identify lining epithelium. Look at it now. You look at the lining epithelium and compare it now here. Identify organ. Identify lining epithelium. That's it. Okay, class, we're ending our lecture, including the liver and the gallbladder, which is supposed to be lectured in 1825, I suppose. I'm not sure. Is it 25, President? Is it 25? But we are done. So you will have no lecture on the 25th. However, I shall inform you that we might have, well, what do you, well, a review or an quiz, but this time it will be submitted, okay? This time, what you have answered, please keep it, you correct it yourself if you got all the layers, if you got all the cells, okay, and the important structures, then you are okay. Well, check it out so that, because that would appear, I'm sure, in any, whether it is in the periodical or in the third bi-monthly or even in the finals, in the gastrointestinal. That's why we have lots of hours for the gastrointestinal tract. So please study the layers of the specific structures, the functions, the name of the characteristic cells. So with that, I'm ending up my lecture. Okay, I will just notify your coordinator. Okay, and whatever, if there is a change in my next lecture. Okay, thank you.
Thank you, Doc.
Thank you, Doc.
Yes.
Thank you,
Thank you,
Doc.
Doc.
Thank you, Doc.
Thank you,
Check
Doc.
your
Thank you, Doc.
check your
Thank
quiz,
you,
your
Doc.
our review.
Thank you.
Check those structures, because
God
if
bless,
you know
Doc.
the organ, you know the structures that are found, you know the functions of the cells, then you know already your entire gastrointestinal, all this parts of your, yeah, of the gastrointestinal. Okay, and then please take note. Remember that your layers in the gallbladder, it is in your book, clearly illustrated. You have only your mucosa, you have your uh, fibromuscular, the third layer, okay, you have only 4, no? And then the perimuscular layer. And of course, either tunica adventitia or tunica serosa, depending where it is attached. Okay, so good luck, and hopefully you will make it to the second year. Bye bye. Thank you.
Thank you, Doc.
Bye bye.
Doc.
Thank you, Doc.
Wala pa ba tayong news about face to face?